a revolutionary concept that reshaped our understanding of cell membrane structure. This model was proposed by two brilliant scientists, S.J. Singer and Garth L. Nicholson, who sought to unravel the intricacies of the plasma membrane of animal cells. Their model paints a vivid picture of the plasma membrane as a mosaic, filled with diverse components such as phospholipids, proteins, cholesterol, and carbohydrates. These components are not static, but rather in constant motion, lending a fluid character to the membrane. As we delve deeper into this fascinating model, we will explore each of these components in more detail, appreciating the complexity and dynamism of the life-giving barrier that is the cell membrane. Ever wondered what makes up the ultra-thin, yet incredibly vital barrier that separates the inner workings of a cell from the outside world? Welcome to the fascinating world of the fluid mosaic model of cell membranes. Imagine a vast ocean teeming with diverse elements that are constantly in motion. This ocean represents the fluid mosaic model, a concept that revolutionized our understanding of cell membrane structure. It is a mosaic filled with different components such as phospholipids, cholesterol, proteins, and carbohydrates, and is fluid given its dynamic and flexible nature. The primary fabric of the membrane comprises amphiphilic phospholipid molecules. These dual-loving molecules have a hydrophilic or water-loving head that interacts with the aqueous fluid both inside and outside the cell. They also have hydrophobic or water-hating tails that prefer to stay away from water. This unique structure allows phospholipids to form an excellent lipid bilayer cell membrane, separating the fluid within the cell from the fluid outside. Proteins are the second major component of plasma membranes. Integral proteins, completely integrated into the membrane structure, interact with the hydrophobic region of the phospholipid bilayer. These complex proteins can span from one side of the membrane to the other, exposed on either side, or associate with a single layer. They are often folded and embedded in the membrane, with their hydrophilic regions protruding from the membrane and in contact with the cytosol or extracellular fluid. Carbohydrates, the third major component, always reside on the exterior surface of cells. They are bound either to proteins, forming glycoproteins, or to lipids, forming glycolipids. These carbohydrate complexes help the cell to recognize other cells and certain substances. To summarize, the fluid mosaic model of the cell membrane is a beautifully complex and dynamic system. The amphiphilic phospholipids form a lipid bilayer, acting as the primary fabric of the membrane. Proteins embedded within this lipid bilayer play various roles, spanning from one side of the membrane to the other. Carbohydrates attached to proteins or lipids on the exterior surface of the cell help with cell recognition. Together, these components create a fluid, ever-changing mosaic that is vital for cell function and life as we know it. So the next time you imagine a cell, picture not just a simple static sphere, but a bustling metropolis encased in an intricate, dynamic, and beautifully complex membrane. Remember, this is not just a barrier, but a gatekeeper, a communicator, and a protector all rolled into one. The fluid mosaic model truly is a masterpiece of nature. As we wrap up our deep dive into the fluid mosaic model, we hope you've gained a deeper appreciation for this masterpiece of nature. It's not just science, it's a testament to the complexity and beauty of life itself. If you've enjoyed this journey and want to continue learning with us, please consider subscribing to our channel. Your one subscribe motivates us to keep creating educational content like this. Remember, every cell in your body is a bustling metropolis and you're the mayor. So keep exploring, keep asking questions, and most importantly,